Hi there, my name is Robert Purrier, and I'm the Chief Scientific Officer for Impact USA. We are an HPLC column company, and today we're going to be talking about ADCs, antibody-drug conjugates.
So what are ADCs? Basically, they're a way to, instead of giving very toxic drugs to the entire body, we only send them to a specific target tissue, like a cancer cell, for example. And we do that by attaching it to an antibody. Now, have you ever seen those commercials where the drug ends in MAB? Guess what that might be? There's several different groups that we see in this field. You could be working on the antibody itself. You could be working on how that antibody and the reaction to attach the drug to it. You could be looking at the efficacy if you're part of a DMPK group. You could also be testing the raw material if you get some products from a third party or even the surfactant. And there's two main types of chromatography used, the purification work, like with SEC, and then the analytical chemistry, which typically uses a protein column, which is a wide pore C4, or a standard pore size C18, for example, if you're looking at surfactants, where you have to do a protein precipitation first. So let's talk about some of the challenges. For example, with SEC, there are some real challenges here. Most SEC columns are mostly polymer based, which means they have low mechanical strength. They can swell, can change if you change solvents. They're not made to work with volatile salts, so not mass spec friendly. And they come in large particles, so very low efficiency, and they cost quite a bit of money. Well, we made a column called Entrada SEC that solves a lot of those problems. It's a high efficiency, 3 micron particles, silica based, and it's designed from inception to work with mass spec. See, here's an example right here. So no longer do you have to tolerate low resolution SEC. You can get high resolution SEC with this column. And I just wanted to show some antibodies and some IgGs. They work very well in this. They're going to work beautifully for your intact antibody analysis. So the next topic is for analytical chemistry folks. If you're using a wide pore C4, I want to talk about why that may not be the best solution for your intact antibody analysis. An intact antibody is about 150 angstroms by 160. I mean, very quickly you can see that 300 angstrom pore size just is not big enough. So our solution is to use a non-porous column, like this Presto FFC18. It's so much better. Here's an example on a typical protein column. It's a C4 wide pore. That's an intact IgG2. And you can see it's a pretty decent peak shape. But on Presto, you can see it's much higher resolution. So why not try that? Give that a try. I think that's a much better option. So the third challenge we're going to take a look at is what do you do when you're doing protein precipitation and using a regular pore size C18? Well, why do that? We make this column called Cadenza HSC18, and it works beautifully. You don't have to do any protein precipitation. It not only saves you time, but waste in the laboratory. Imagine all those vials you're throwing away to do protein precipitation. And your data might be better because it's one less variable to have to control for. And how this works is it has one additional ligand, and this additional ligand essentially creates a force field around the particle so that the proteins just bounce right off, wash off the column, while still retaining your small molecules like surfactants, just as any other C18 would. And here's an example chromatogram of that. You can see the tween 20s retain nicely, but all those antibodies come out completely unretained. And in fact, there is a group at Amgen that wrote this paper. I highly recommend you take a look. And they tested several different columns. They even did quite a bit of method development work. It's a wonderful paper to take a read. But their conclusion was that Cadenza HS was by far the best solution.
Well, thanks for watching. I hope that it was helpful. This is, again, such an important field, but the chromatography can be quite challenging, especially since we're dealing with molecules that are 150 kilodaltons or even larger. I hope now you understand that we make some great products that can definitely be a benefit to you in your lab. You know, our Entrada SEC for high resolution MS SEC work. We also have the Presto FFC18, that 2 micron non-porous column that has no size limit. It can easily handle 150 kilodalton antibody. And of course our Cadenza HSC18, that wonderful direct injection column. It's very reproducible. Whether you're trying to do protein depletion work to uncover what's going on with the surfactant or the unbound small molecule, you know, that's a fantastic product. If you have questions about this or anything else, please hit me up. I'm at info@mtacusa.com and I'd be happy to work with you.